My name is Brandon Guell. I'm a student at the University of California in San Diego, and I'm going into my 4th year, and my major is ecology, behavior, and evolution biology, and I'm minoring in chemistry. I was lucky enough to have an older sibling, so the whole application process and visiting schools, I kind of tagged along for that. But in looking at what I had already seen and in going through the application process and looking at different schools, I was really interested in going to an R1 university, which is emphasizes research and has a lot of opportunities for undergrads to get involved in research. So that was definitely one of the things I was prioritizing and looking at a university. Also location and biology programs in general. So UCSD was actually a great fit for me because I love the location, I love the beach, it's close enough from home, and I have an outstanding research program for biochemistry, ecology, chemistry, physics, math. A bunch of their departments are really known for having great research programs. I think in high school too, you have a really close-knit group of friends that you study for, especially if it's AP tests or just for exams in the classroom. You definitely have a smaller group of people that you study with normally, especially in the higher level high school classes. And so just kind of feeding off each other's motivation and interests was really an awesome experience too, because in college you kind of are more independent. So having that in high school was really important for me to see that wow, there are other people that are interested in biology, and it doesn't have to be one specific thing about biology, because there's ecology, there's biochemistry, there's cell biology, there's ecosystem biology, there's a bunch of different types. And so kind of growing in a little cohort.
That makes perfect sense.
Yeah. So that was a nice segue into kind of seeing what I really enjoyed about biology and then pursuing that in my college career. So I was really fortunate to actually have teaching assistants in upper division biology classes that were looking for assistants in the lab. So I started off cleaning and doing a bunch of microscope work, which is not that interesting, but it kind of gets your foot through the door. And so taking that first step was really important, because after that it opened up so many other doors for me. I ended up doing an awesome research experience last summer through the same lab and working with PhD students and professors and getting really close connections and kind of networking and understanding and experiencing how research works and just getting a taste of that. And eventually that led me to applying for an REU this year.
What is that again, REU?
So an REU stands for Research Experience for Undergrads, and it's an NSF, National Science Foundation funded program that is usually a 10-week program at either a university or an institution that emphasizes or that has undergrads experience research in a bunch of different fields. And so coming to STRI was really awesome for me because ecology.
What's STRI?
So STRI is the Smithsonian Tropical Research Institute in Panama, which is where Karen Workington's lab is, who I'm interning for this summer through the REU program. So that's it was an awesome opportunity for me because I'm really interested in ecology and evolution, and this is a fantastic place to do it. And studying frog embryos is one of the coolest ways to do that.
Awesome.
So our lab looks at developmental changes in one species of frog and how those developmental changes influence premature hatching and embryo behavior when exposed to environmental cues. And so what I'm looking at specifically is how development affects embryo behavior and hatching performance. So there's a lot to know about this specific frog and these embryos, but the most important is basically that they hatch within 6 or 7 days upon being laid.
What species is this?
So we work with red-eye tree frogs, which is species name is Agalichnus calidrius, and they're a neotropical tree frog that lay gelatinous eggs on leaves and other substances that overhang ponds. And so when it rains a lot, the frogs go out and they mate and they lay these clutches of eggs over the pond. And so typically on the 6th or 7th night, after being laid, the frog eggs hatch normally, But there's a lot of things that can go wrong or that can happen that might force them to make decisions to hatch earlier, right? So they're preyed upon by snakes and wasps. They can be attacked by fungal infections. They can be flooded if there's a heavy rainfall. And so throughout development, the embryo has to make decisions on whether this cue that they're getting, whether it's vibrational, visual, is dangerous or if it's benign. So if it's rainfall, they can sense that and feel that, and they have to determine if that's different from a snake vibration while it's attacking. And so what I'm looking at specifically is looking at how development affects their choices in hatching or not, and how well they hatch out of their eggs. So how do they know to hatch out the front? How do they know how to hatch when one of their environmental like how do they know how to hatch when they're flooded and there's no oxygen gradient, which they usually use to position themselves in the front of the egg? Like what other cues can they be using in these special circumstances, basically, to know when to hatch and how to hatch? So based on some observations in the lab on previous experiments, we noticed that the embryos might be using some type of visual cue to know how to hatch and where to hatch. So what this means is, so what we wanted to test is to see if the embryos were using light as a source of information to basically hatch in the direction or in the opposite direction of where this source was coming from, this light source is coming from. And so what we did is we built these little cups that basically had a plastic tube in the middle where the egg could go inside, and half of the cup would be completely dark and the other one would be exposed to light. In order to basically make them hatch, we flooded them in anoxic water, so there was no oxygen, which causes them to basically hatch out of their eggs. And then it's like a choice test, so they can either go towards the complete darkness or they can go towards the light. And so this would hopefully give us a better understanding of whether they're using this visual cue in order to decide how to hatch and where to hatch when under these like anoxic conditions.
Are you still carrying out that experiment now?
So that experiment is done, and what we found is that there is a positive phototaxis, which means that the embryos were hatching towards the light. So we were really excited to see that because we hadn't previously known that there was any type of visual cue that the embryos were using in order to make decisions on how to hatch and where to hatch.
So you, as a fourth year, between your third and fourth year undergrad student, was able to make a pretty significant finding in that these eggs do use light cues as far as which direction they hatch, yeah?
Yeah, I mean, I like to think that it's pretty significant, and even the simplest little question can just be like really interesting, and it kind of just provides a better understanding of information use in animals and developmental changes, and it kind of just contributes to the larger picture idea of looking at plasticity, so how the animals behave differently throughout their development when exposed to experimental cues.
Excellent.